Good afternoon, student. I am Naneshwar Pawar. Welcome to everyone's in Enlightenment Agri Academy. So today we will be discuss the how to crack the plant pathology ASRB NET and ARS 2020. So here we will discuss the complete syllabus breakdown and strategy for this examination. So firstly, we will see the overview of this examination. The full form is the Agriculture Scientific Recruitment Board, which conduct these examinations. Then what is the purpose of this examination after you qualify for NET ERS? So if you qualify for NET, then assistant professor in agriculture university. When you qualify prelims for ERS, then you get mains examination, then interview, and then you select as agriculture research scientist in agriculture research station or institute, and also for the some subject matter specialist as a plant pathologist in some institutes. So here conducting body is the National Testing Agency conduct this examination, and this examination frequency in once in a year expected in November 2020-26. So here eligibility criteria for this examination for educational qualification, that is the MSc in plant pathology is needful for this examination, and ICAR accredited college is. So age limit for this examination is the minimum 21 years, or upper age limit there is no limits for this examination. Now the nationality is a nationality is merged with a Indian. So now exam pattern for this examination: computer based test, then 2 hours for this examination, ask the 150 questions, MCQ questions, medium is English. Marking scheme for the correct answer is 1 mark, for incorrect answer minus 0.25 marks will be negative marking. So unattended answer there is no negative marking, that's the total 150 marks. For the qualification marks, you need to, for general category, nearly about the 50% mark. For OBC category, nearly about the 45 marks. And for SC, ST, nearly about the 40% mark. But the year-wise cutoff will be changed due to the pattern of examination, then difficulties of the examination. So here is not a fix, but have the some idea about that qualifications. Now the ASRB NET, ARS, STO, SMS, plant pathology syllabus unit. So today we'll discuss the units, total 11 units, and now discuss the one by one in next slide. So here we overview. First unit have the history and principle of plant pathology. In second unit, laboratory and analytic techniques. Unit third, physiological and molecular plant pathology. In fourth unit, mycology. In fifth unit, plant bacteriology. Sixth unit is the plant virology. Then seventh unit, disease epidemiology. Eighth, phanerogamic parasites and non-parasitic diseases. Unit 9 is the fungal diseases of crop plants. 10th unit, bacteria and viral diseases of crop plants. And 11th unit, the management of plant diseases. This one is the most important for all the plant disease control. So now discuss one by one their breakdown of the syllabus, how to study and how to need to study. So here unit first, history and principle of plant pathology. So here first one is the milestone, the how the observation was taken by the earliest scientists in the India and also for the all over the world. So there is observation how they how to identify the disease, how disease was caused, how to prevent, cure, and how to prevent the disease. So second one is the major epidemics and their social impact. How the epidemics will impact on the social economy, food security, and huge crop losses. Now the example we will taken 1845, the late blight of potato was famine in Irish, so that's why the 1 million people were died. In India also in 1945, there is the Bengal famine due to Helminthsporium oryza, which caused in the rice. So that's why how the low huge losses on farmers and also for the food scarcity on that time. So historical development for chemical, legislative, cultural and biological protection. Now the government of India take the some controls protection for the disease, disease control. Now the classification of plant diseases have the on the basis of the fungi, bacteria, viruses and nematodes. So we will discuss one by one in our upcoming lectures. So physiological specialization, what the physiological specialization present in the fungi, bacteria, viruses, they have the some spatial characterization to live long-lasting period in the soil or any debris of the plants. So now the course postulate was find the three course postulate stages. First one is they found, then it isolate, then infect to the plants, then re-isolate, and fourth one is the then once again re-isolate. So this fourth one is given by E. Smith. Now the growth, reproduction, survival, and dispersal of plant pathogen. So factor influencing infection, colonization, development of the symptoms. There is the for the factor influencing the infection, colonization, and development of symptoms. There is a triangle between the host, pathogen, and environment. Now here is required the pathogen is pathogen is have the ability to cause the disease. That means have the higher pathogenicity. Now the host is susceptible to the pathogen. Now the environment, that is the favorable environment need for the disease development. Now the second unit we will discuss here laboratory and analytical techniques. So preparation and sterilization of common media, how the media will be prepared, how they autoclave, how the dry oven, hot air oven, uh, glass vase will be sterilized, and then liquid and filters. So these are the methods will be discussed in our upcoming lectures. Then method of isolation of pathogen and their identification. How will be the identify found the stick? That means single colony will be isolated. That is the stick. Then pouring, the total mix culture will be the pour, and dilution. Now the dilute the for the pure culture isolations. Now the single bacteria or fungi isolation. So now the morphological, cultural, biochemical, and molecular identification. Firstly, we will identify on the morphology, then basis of the culture, then biochemicals, and then molecular characterization will be done, and then will be identify this microorganism which cause the disease. The preservation of microorganism in pure culture, how they preserve? First one, they make the stands, then oil, then freeze, and then dry. Have some more method to preserve, but one by one we'll discuss. So method of inoculation, how the inoculation will be done by the mechanical. That means the rupture the plants leaves, and then incorporate this inoculum into the plants, then through the vectors and through the soils or seeds. Then measurement of plant diseases, how the plant measurement diseases through the incidence, severity, and have some score was given by some scientists. And now on that basis we will be measure the plant disease impact. So now the molecular detection of plant pathogen in seeds, other planting material, nucleic acid probe, southern, northern, and western hybridization, ELISA test, then ESM and PCRs. So laboratory equipment for their uses. Which laboratory equipment is for this procedure? Now the autoclave. What is the role of the autoclave? Sterilization of the liquid media. Then for the hot air oven, sterilize the glassware, laminar airflow, then photophotometer, electrophoresis, light electron microscopy, incubator, then ultracentrifuge, ELISA reader. So these are the equipment will be used in a laboratory. So here discuss the third unit, that physiological and molecular plant pathology. The alter the metabolism of plant under the biotic and abiotic stress, molecular mechanization of plant pathogenesis, then recognition phenomena, penetration, invasion, primary disease development. How this how did the disease development was a mechanism developed in the plants? We will discuss. So enzymes and toxins in relation to the plants, then mechanism of resistance, what is meant by phytoalexin, what is meant by PR proteins, what is meant by antiviral proteins, what is meant by SAR, systemic acquired resistance, what is meant by HR, then active oxygen radicals, and how they use in a disease resistant mechanism. Now the tissue culture also play a very important role in the development of the virus-free plants. Now the somaclonal variation, what is meant by somatic hybridization, elementary engineering. Then management of pathogen through the satellite, antisense, RNA, ribosome, code protein, then hypovirulence, cross protection, useful genes, and promoter technology, biosafety, and bioethics. Then unit fourth is the basic unit, have the most weightage as compared to other units for the NEET examination and also for ARS examination prelims. So here the classification of fungi classify based on the spore, based on the sexual reproduction, based on the structure. Now their economical uses, some are the edible fungi, then entomogenous fungi, Bavariana bassiana, Metarhizium anisophilia. So now these are the beneficial for the endogenous purpose. Now industrial aspergillus is produce the citric acid. Then once upon the penicillium. Penicillium is also useful for the make the antibiotic penicillin. Now the mycorrhizal association, mutualism relationship between fungi and plants, and how they uptake the phosphorus, how they act as a disease control agent in some diseases. So there will be importance. Now the cell organelle, their morphology, function and chemical composition of fungi. So here the nucleus, what is the nucleus, ribosome, how they produce the proteins, how the mitochondria give the energy to the cell, then endoplasmic reticulum functions, then Golgi bodies, lysosome, plastid, that means chloroplast. Now here discuss the fifth unit, that is the plant bacteriology, the identification and classification of bacteria, morphology, their shape, staining, structural features, and other classification. Now on the basis of the shape, there are the cocci, a rod shape, then urula. These are the some shapes. Now the morphology, ultrastructure, chemical composition of prokaryotic cells in relation to functions. And then what is the growth curve, how the lag phase, long phase, stationary, then that nutrition, exotropic, Newton. Then resting cells in a prokaryotes, how the bacillus have ability to produce the endospore, and that endospore is resistant to resistant to environment, and now they live long lasting period for the thousands years. Now the elementary bacterial genetics and variability, transformation, conjugation, transduction, now the transformation done by the viruses, conjugation between the direct contact between the two bacteria, and how the transduction will be done. So now we study the biology of extra chromosomal element, plasmid borne genes and their expression, EUR gene, HER gene, UIE gene, then PAT genes. So how the genes will develop and the impact on bacteria. So bacteriophages means what is meant by bacteriophages? They replicates. Then outside the uh, bacterial cell, viruses were replicated, then host cell lysis, and then new phase was released. In the lysogenic phase, DNA is integrated into the host genome and dormant until the triggered condition. For the favorable condition, they will be dormant on these plant cells. Now the prokaryotic inhibitors and their mode of action: penicillin, penicillin, then tetracycline, rapamycin, then polymyxin, then economic uses of prokaryote, food industry, agriculture, morphology, chemical characteristic, reproduction, life cycle of phytoplasma and other fastidious prokaryotes. Now the sixth unit is the plant virology. What do you mean by nature, composition and architecture of viruses and viroids? What is the property of the virus? They are the obligate parasite. They only feed on the living organisms, and they also some cause the diseases. They transfer through vectors. Now they transfer through vectors. So variability in viruses, satellite. What is meant by satellite viruses? How the satellite RNA develop? Then assay of the plant viruses, including the detect and quantifying viruses using the biological, physical, chemical, and serological and molecular method. Then conventional biotechnological techniques used in detection and diagnosis. Behavior of viruses of plants, including infection, replication, movement. Then histopathological changes included by the virus in plant inclusion and bodies. So transmission of viruses, how the virus was transmitted to the use of the vectors, how the nomenclature of the viruses, their classification, and nucleic acid type, symmetry, envelope, host, and ranges. So this is the 6th unit syllabus. So now we will discuss 7th unit, that is the plant disease epidemiology. So concept of the epidemiology, what is meant by epidemiology, what is meant by disease triangle, we will already discuss host pathogen environment for the disease triangles, then development of diseases in plant population, then a disease progress curve, incubation period, then latent period, epidemic phases, decline. Then what is meant by monocyclic, what is meant by polycyclic pathogens, the role of environment and meteorological factors in development of plant diseases, what is meant by epidemics, disease severity depends on temperatures, humidity and rainfall, winds. So survey, what is meant by survey, go to the fields and take the samples and identify the diseases. So this is the sample survey. What is meant by surveillance, including to the remote sensing and prediction and forecasting of diseases. Then epidemic analysis and prediction model. The logistic model, compares model, vendor plan model. So these are the epidemic analysis model, three models. So the crop loss assessment, critical and multiple point method. So in this plant disease development, these are the basic topics, and we will discuss about the more in our upcoming lectures. So we will be discuss the 7. Now I will discuss 8th unit. So here the 8th unit, the phanerogamic parasite and non-parasitic diseases. But this is caused by the phanerogamic parasites and their management. There are the two types of, the first one is the root parasite, stem parasite. Then again they classify into the two, when the semi-root parasite, then partially and full parasites. Then there is also classified into two types. This is due to the unfavorable soil environment, drought, flooding, trace causes, but in the flow, poor drainage, then salinity, extreme pH, waterlogging. This is the cause. This is unfavorable environment in the soil. Now the nutritional deficiency, through the due to the deficiency of nitrogen, the yellow, the older leaves. But due to the ferrous deficiency, will cause the yellow young leaves. So this is the difference between the two nutrients deficiency. Now the primary, secondary hail pollutant, acid rain, primary hail pollutant such as the sulfur dioxide, nitrous oxide. Then secondary is the uh, ozone layer, acid rain, leaf plus soil damage. How the acid rain? So as the impact on the leaf and soil damage. So here we will discuss the unit 9 is what is meant by the fungal diseases of crop plant. The fungal diseases are the cereal, millet, oilseed pulses, fruits, vegetable, plantation, fibers, species and ornamental plants. So etiology, what is meant by etiology? How the disease cycles of the this pest diseases, how the perpetuation, then epidemiology and management. So these are the basic concept in this regarding to the fungal diseases. Uh, then post-harvest diseases in transit storage. What is meant by alpha toxin? Or alpha toxin by produced by aspergillus present on the groundnut. So these are the impact on foods of the humans and also for the animals. So we will discuss the 10 unit, that is the bacteria and virus diseases of crop plants. The crop diseases of the cereal, pulses, oilseed, vegetables, fruit, plantation and fiber crop. Now the bacteria we will discuss the wilt, blight, canker and is spread via the water, tools and soil. Viruses, mosaic leaf curve were spread by the insect and vectors. The viroids will spread mechanically, vegetative propagation, then have the symptoms like the stunting, deformation. Then phytoplasma, yellow, little leaf, spread by the leaf hooper. Then management mantra. For this, use the resistance variety, use the control vectors. So need to spread the insecticide, then sanitization, chemical, mode of transmission, and pathogen vector relationship, and how it is the epidemiology and how it managed. So now we'll discuss the last unit, our 11 unit in this lecture. So this one is the management of diseases, management of plant diseases. Now the general principle of plant quarantine. What is meant by quarantine? How it is need? Some pathogen will become in our region, so that's why plant quarantine is needful. Then how the infection will be done, how the plants isolate, then how give the certification by the certifying body. So this one is the basic concept. So exotic plants and pathogen introduced into India. Exotic pathogen in India, examples like the Puccinia gramineatis comes from another country. Xanthomonas oryzis also from another country. Bacterial blight in rice. Then sanitary and phytosanitary issues under the WTO, TRIPS, PRA. So this one is the issues will be discussed on these topics. So genetic basis of disease resistance, pathogenicity. What is meant by pathogenicity? What is the gene for gene hypothesis? Was given by Fleur. So parasite mediated frequency dependent selection concept of QTL mapping. Then breeding of disease resistance, what the R gene, what is meant by AVR gene, use the pyramid for the durability. So here the production of disease-free seeds and planting material, and give the certification to the certifying seed certification body. Then chemical nature and classification of fungicides, antibiotics, how the classifiers on basis of the systemic fungicides, uh, contact fungicides, the antibodies like the penicilliums and some other antibodies. Then use their bio as a compatibility with the other agriculture chemical resistance to fungicide antibiotics, or effect on environment, spraying and dusting the equipments, spraying dusting equipments, their care and maintenance, how they maintain, how their care will be taken, how to spray and dust. So this one is important. The important culture practices and their role in disease management. Solarization means, what is meant by solarization? What is meant by integrated disease management? In the integrated disease management, we will be use the culture method, we use the mechanical method, we use the chemical method, we use the biological method. So this is the firstly the objective of the integrated disease management is here, and then disease will be not come before their maximum infestations. Control disease below the threshold level. That is the main purpose of integrated disease management. So now the microorganism antagonistic to plant pathogens in soil, rhizospheric. What is meant by rhizosphere? What is meant by phyllosphere? And their use in the control of plant diseases. Then what is meant by fungus soil? Fungistat. Fungistat means is uh, some uh, some fungi have ability to reduce the growth of the other organism. So that's called as the fungicide. That means not the kill, but only the inactive. So that's why fungicide is. So what is the plant growth promoting rhizobacteria also play important role. Now on the plant growth promoting rhizobacteria, need to research on that growth promoting rhizobacteria for the control of diseases. So thank you student. We will discuss here about total 11 unit. In this lecture, we will discuss the 11 units about the ARS examination. And now you like, share, and subscribe our YouTube channel, and comment your doubts on comment section. So then I will be reply on that comment. So thank you here.